2 million years ago, in a limestone cave 40 kilometers northwest of what is now Johannesburg, something with huge molars and a face built like a nutcracker died and left its teeth behind. Nothing else about that moment mattered to science for the better part of a century. Bones fossilize. Genetic material does not in Africa, not across that kind of span, not according to everything researchers had assumed for decades. And yet in 2023, and again in a more complete study published in 2025, a team of researchers pulled something out of those exact teeth that should not have survived. Not a full genome, not quite DNA in the way the word usually gets used, something adjacent to it, something that had never been recovered from anything this old on the African continent, and something that, when they finally read what it said, didn't just answer the question they'd gone looking to answer. It opened up a new one, a lineage sitting inside a species that already had a name, quietly not matching it. To understand why this matters, you have to first understand a strange and slightly embarrassing gap sitting at the center of paleogenetics, the science of reading ancient genetic material. Ancient DNA has completely rewritten human prehistory over the last 15 years. It's how we know Neanderthals interbred with the ancestors of every non-African population alive today. It's how we know an entirely separate group of archaic humans, the Denisovans, existed at all, identified initially from nothing more than a finger bone and a single tooth found in a Siberian cave. It's how researchers have traced population movements, kinship networks, and migration routes across tens of thousands of years with a level of precision that would have seemed like fantasy to an archaeologist working in the 1980s. Almost every one of those breakthroughs, though, shares one thing in common. They come from cold places. Siberia, Northern Europe. The kind of environment where DNA, an inherently fragile molecule, gets a fighting chance against time because the cold slows the chemical processes that tear it apart. Africa is where the human story actually begins. Every early hominin lineage, every branching point on the family tree that eventually produced us, unfolded there first, for millions of years, before anyone ever left the continent. And Africa, for exactly this reason, is the single hardest place on Earth to recover ancient genetic material. Heat and humidity are exactly the conditions DNA degrades fastest in, and most of the African continent has been warm and often humid for the entire span of time relevant to human evolution. Ancient DNA has been transformative for studying more recent relatives like Neanderthals and Denisovans, but it doesn't preserve well in Africa because of its simple molecular structure breaking down under warmer conditions. That's not a minor technical footnote. That's the single biggest asterisk hanging over the entire field. The place where the most important part of the human story happened is, for genetic purposes, close to a black box. Researchers have spent decades reading the story of human evolution almost everywhere, except where most of it actually took place, which is what makes the specific find at the center of this story worth taking seriously. The fossils came from Swartkrans Cave, part of the cradle of humankind, a UNESCO World Heritage Site whose limestone caverns have produced some of the richest hominin fossil records in the world. A team of European and African researchers retrieved protein fragments from the dental enamel of 4 fossil teeth using a method called paleoproteomics. The teeth belong to Paranthropus robustus, sometimes nicknamed Nutcracker Man, in older textbooks for its enormous jaw muscles and thickly enameled molars built for grinding down tough, fibrous plant material. Robustus lived across a stretch of southern Africa roughly between 2.5 million and 1 million years ago, meaning it coexisted for a significant span of time alongside the earliest members of our own genus, Homo. It was not our direct ancestor. It sat on a side branch, a cousin lineage that eventually died out entirely, leaving behind nothing but fossils and, as it turned out, something considerably harder to find hidden inside them. The workaround the researchers used deserves its own explanation because it's genuinely elegant, and it's the reason this discovery became possible at all. DNA carries genetic instructions directly, encoded in a sequence of 4 chemical bases. Proteins are what genes actually build, the working molecules assembled according to those DNA instructions, and their building blocks, amino acids, are considerably more chemically stable than DNA itself. While DNA carries genetic information, proteins are the expression of that genetic information. And because the amino acid sequence of a protein can be reverse engineered by scientists, information about the underlying DNA can still be recovered, even after the DNA itself is long gone. Proteins survive dramatically longer than DNA under harsh conditions. Some recent paleoproteomic work has pulled readable protein sequences out of animal teeth dated to as much as 21 to 24. Million years old, recovered from an ancestor of the modern rhinoceros, preserved in the frigid conditions of Canada's high Arctic, and almost unthinkable span when measured against DNA's usual limits. Tooth enamel turned out to be an especially good vault for this kind of preservation because it's the hardest, most mineral-dense tissue in the entire human body. dense enough to lock certain structural proteins in place and shield them from the enzymes and bacteria that would otherwise dismantle them within centuries rather than millennia. The technique itself relies on an instrument called a mass spectrometer, which doesn't read genetic sequence the way modern DNA sequencing machines do. Instead, it measures the precise mass of tiny fragments of protein, and from those masses, researchers can work backward to reconstruct which amino acids were present and in what order. It's an indirect method, more like carefully reading a shadow to reconstruct the shape of the object that cast it than reading the object directly. It requires state-of-the-art equipment, painstaking sample preparation to avoid modern contamination, and enormous statistical care, because the fragments recovered are short, damaged, and incomplete. Nothing like a clean, continuous strand. When Enrico Cappellini's team at the University of Copenhagen first applied this approach to the four Swartkrans teeth, the initial results posted in 2023 and expanded in a fuller 2025 publication with colleagues including Palisa Madupe, Claire Koenig, and Ioannis Patramanis were described by the researchers themselves in fairly measured terms. It was described as the earliest genetic information ever gathered about any ancient hominin, extracted from 4 Paranthropus robustus teeth found at Swartkrans, with roughly 400 amino acids ultimately sequenced across the samples. 400 amino acids sounds modest against the scale of a full genome, which runs into billions of base pairs. But this wasn't a full genome. This was a fragment, a keyhole, and even through a keyhole this small, several things became visible that had never been visible before. The first thing the proteins revealed was something almost startlingly simple, and yet nobody had ever been able to confirm it directly for fossils this old: sex. In 2 of the samples, researchers detected a melogenin Y, a protein produced by a gene located on the Y chromosome, confirming those individuals were male, while the other 2 teeth carried the X chromosome variant of the same protein instead, indicating they came from females. Sit with that for a moment, because it's easy to read past quickly. For over a century, paleoanthropologists have had to guess at the sex of hominin fossils, almost entirely from bone size and robustness, a method that is genuinely useful, but also genuinely uncertain, especially in a species like Paranthropus robustus, where scientists have long debated whether size differences between specimens reflected sex differences within one species, or whether they might actually reflect entirely separate species being lumped together under one name. Molecular sexing, done directly off a 2-million-year-old protein fragment, is a different order of evidence entirely. It doesn't guess, it reads. The second finding is the one that actually reshapes the story, and it involves a protein called enamelin, which plays a central structural role in how tooth enamel forms. The analysis of protein sequences revealed subtle but potentially important genetic differences among the fossils, centered on a variation in the gene responsible for producing enamelin. 2 of the 4 individuals carried one version of this protein, an amino acid variant also, found in modern humans, early humans, chimpanzees, and gorillas, essentially the common ancestral version shared broadly across African great apes. The other 2 individuals carried a different variant, one that, among the African great apes studied so far, had only ever previously turned up in Paranthropus. On its own, that split might have looked like exactly what researchers initially assumed: a marker unique to the Paranthropus lineage as a whole, something that simply defined the genus. But the team found that what they had first thought was a mutation unique to Paranthropus robustus was actually variable within that same group, with some individuals carrying it and others not, marking the first time a protein mutation of this kind had been documented in ancient proteins rather than ancient DNA. That single sentence is where the story stops being a technical triumph and starts being genuinely strange. If all 4 teeth belonged to the same species, and if this enamelin variant were simply a species-defining trait, every individual should share it. They don't. 2 carry the version everyone expected. 2 carry a variant that looks like something else entirely, something that resembles a genetically distinct population rather than ordinary individual variation. Researchers concluded these results suggest that what appears to be a single, variable species might instead represent a more intricate evolutionary pattern involving individuals with genuinely different ancestries. In plainer terms, the fossils that have been filed under the single name Paranthropus robustus for decades may not all belong to one tidy, uniform population. Buried inside that name may be at least 2 genetically distinguishable lineages, coexisting, perhaps interbreeding at the edges, perhaps not, in the same caves at the same time, and nobody currently has a name for the second one. It has never been formally described as its own taxon. It doesn't show up in any museum placard. It exists right now only as a pattern in a handful of amino acids recovered from 4 teeth, waiting for enough additional evidence to justify actually naming it as something distinct. This is not a small taxonomic footnote either, because it lands directly on top of a debate that paleoanthropologists studying Paranthropus have been having, inconclusively, using only bones and skulls for the better part of 50 years. Researchers studying this material have long asked whether the notable size differences between specimens reflected simple sex differences within one species, or whether they instead pointed toward the presence of multiple distinct species being grouped together under a single name, along with open questions about how Paranthropus robustus related to other hominins and to early Homo, and what, at a genetic level, actually made it distinct. Morphology, the study of the physical shape of bones and skulls, had taken that argument about as far as it could go and essentially stalled there because skull shape alone can't cleanly separate these are two sexes of one species from these are two closely related but separate species that happen to look somewhat similar. Until this protein study, answers to those exact questions had remained elusive, in part because ancient DNA, the tool that had already revolutionized the study of more recent hominins like Neanderthals and Denisovans, simply doesn't survive well. Enough in Africa's climate to be recovered from material this old. Paleoproteomics didn't just answer the sex question directly. It cracked open a version of the species question that bones alone had never been able to resolve, and the crack it opened doesn't lead to a clean answer. It leads to more questions, with real molecular evidence behind them for the first time. It's worth being precise here about what this evidence does and does not establish, because the honest version of this story is more interesting than an overstated one. 4 teeth is an extremely small sample. The researchers themselves have been explicit that confirming P. robustus fossils actually represent different ancestries will require sampling tooth enamel proteins from considerably more individuals, and that this current result, while striking, is a first indication rather than a settled conclusion. The amino acid differences detected are also just that, differences in the sequence of a handful of proteins, not a full reconstructed genome. A protein fragment can tell you something is genuinely different at a molecular level. It cannot, on its own, tell you exactly how those two lineages are related, how long ago they diverged from each other, whether they interbred, or whether the split is significant enough to warrant a new species. Named under the formal rules of taxonomy, that's a much larger undertaking, and it's one the same research groups have already signaled they intend to pursue by extracting protein data from additional Paranthropus fossils recovered from other South African sites. The finding, in other words, is less a closed case and more the opening of an entirely new kind of investigation, one that didn't exist as a methodological option before this study proved it was possible. There's a broader significance to this that goes beyond Paranthropus robustus specifically, and it's the part of the story that makes this feel less like a single discovery and more like the opening of an entire new field of inquiry into deep African prehistory. Every major turning point in the story of human origins happened on that continent. The emergence of the genus Homo roughly 2.5 million years ago, the appearance and spread of Homo erectus starting around 2 million years ago, the eventual emergence of our own species, Homo sapiens, sometime before 300,000 years ago, all of it played out primarily across Africa long before any group of humans crossed into Europe or Asia. And virtually none of that deep African history has ever been directly readable at the molecular level, because until paleoproteomics matured into a workable tool, the climate made ancient DNA recovery there close to impossible. This study is a proof of concept that the molecular record of Africa's deep past isn't actually gone. It's just been sitting in a form nobody had a reliable way to read yet. That proof of concept is already extending outward from Paranthropus into the genus that actually produced us. In the work reported in the first half of 2026, researchers applied the same enamel protein approach directly to Homo erectus, the species widely regarded as the first hominin to leave Africa and spread across Asia and possibly Europe, with fossils recovered from locations as far-flung as Indonesia, Spain, China, and the Republic of Georgia. Homo erectus arose in Africa roughly 2 million years ago before spreading across the globe, but because genes and proteins preserve so poorly in most of the regions where its fossils have been found, information about its internal genetic makeup had remained largely out of reach until researchers began extracting ancient enamel proteins directly from its teeth. That's a genuinely significant escalation from the Paranthropus work, because Homo erectus sits far closer to our own branch of the family tree than Paranthropus ever did, meaning any genetics signal recovered from it has direct bearing on understanding our own species' immediate ancestry, not just the ancestry of an extinct side branch. Set that finding against the earlier 2023 preprint's own broader conclusion, and the shape of the whole emerging field becomes clearer. Sequencing across all 4 samples produced enough overlapping information to construct a simple evolutionary tree, one that demonstrated Homo sapiens, Neanderthals, and Denisovans living within roughly the last 200,000 years are far more closely related to one another than any of them are to Paranthropus. That result is, on its surface, exactly what anyone would have predicted based on decades of skeletal and archaeological evidence. Paranthropus was always understood to be a distant cousin lineage, not a direct ancestor. What matters here isn't that the tree confirmed the expected shape. What matters is that, for the first time, a piece of actual molecular evidence pulled directly out of African fossil material, rather than inferred indirectly from bone shape or from DNA recovered on other continents, could be plotted onto that tree at all. The researchers behind the preprint described the ability to build an evolutionary tree from genetic material recovered from fossils this old as a potentially transformative development for the entire field of paleoanthropology. That's not an overstatement. It's closer to an understatement of what a working technique for reading 2 million-year-old African genetic material actually opens up. Consider, for a moment, everything that's been sitting undisturbed in African cave systems for the better part of 2 million years, waiting on exactly this kind of technique. Skulls, teeth, and partial skeletons of Australopithecus, the older and more primitive genus that gave rise to both Paranthropus and, eventually, Homo, sit in museum drawers and cave deposits across Ethiopia, Kenya, Tanzania, and South Africa, some of them well over 3 million years old. Early Homo habilis material, Homo naledi, remains recovered from the Rising Star cave system, a species so recently discovered and so unusual in its mix of primitive and modern features that its exact place on the family tree is still actively debated. All of it currently understood almost entirely through bone shape, dating methods, and archaeological context, none of it yet successfully read at the molecular level. If the technique that pulled readable protein sequences out of 2 million-year-old Paranthropus enamel can be pushed further, refined further, applied to a wider range of specimens, there's no obvious reason it has to stop at Paranthropus and Homo erectus. The entire deep genetic record of human origins, the part of the story that has always been the hardest to read directly, may finally be within reach. Not through DNA, which simply won't survive that long in that climate, but through the sturdier molecular echo that proteins leave behind. And that returns us to the strange, still open thread sitting at the center of the Swartkrans discovery itself. A population that shares a name with Paranthropus robustus, that was buried in the same cave, that shows up in the same fossil catalog, and that carries, in a protein nobody had ever been able to test this way before, a signature that doesn't match. 2 teeth, 1 genetic signature, 2 other teeth, a different one, older, rare, previously seen only in the wider Paranthropus lineage as a whole rather than as an internal split. Whatever that second signature actually represents, whether it turns out to be a genuinely distinct species that coexisted with the more familiar form of Paranthropus robustus, a deeper ancestral population contributing to a mixed group, or something else entirely that current sampling is too small to pin down, it does not yet have a name. It sat in that cave for 2 million years under a that may never have fully belonged to it, waiting for a technology that didn't exist until a handful of researchers, sequencing amino acids, 1 damaged fragment at a time out of 4 fossil teeth, finally gave it a way to say so. The honest ending to this story isn't a tidy one, and that's exactly what makes it worth paying attention to. This isn't a case where scientists set out looking for a hidden species and triumphantly found it, complete with a new name and a press conference. It's a case where a team trying to answer a much narrower, more technical question, whether ancient proteins could even survive long enough in African conditions to be read at all, stumbled into evidence suggesting the answer to a much older, much bigger question, what exactly Paranthropus robustus actually was, might be more complicated than the fossil record alone had ever been able to show. The species without a name isn't a headline invention. It's a real gap sitting inside real data, acknowledged directly by the researchers who found it, who are now doing exactly what good science requires, going back for more teeth, more caves, more fragments, trying to find out whether that gap closes into a clear answer or widens into something genuinely unprecedented in the story of where we came from. Either way, the fact that this question can now be asked with actual molecular evidence, rather than skull measurements and educated guesswork, is itself the real discovery. Africa's deepest genetic past, locked for 2 million years inside tooth enamel nobody thought could hold a readable trace that long, has just started talking. It hasn't finished yet.